Number 1. The Yuka Mammoth. We begin in the year 2010 on a stretch of permafrost called Oyogos Yar near the coast of the Laptov Sea in northeastern Siberia. Local tusk hunters searching the thawing riverbank for ivory found something far stranger than ivory. They found a body. Not a skeleton, not a scatter of bones, but an actual carcass with skin still on it. This was a woolly mammoth, and it would come to be called Yuka, named for a nearby Siberian settlement. Yuka is roughly 39,000 years old. The animal lived and died in late Pleistocene Siberia during the last Great Ice Age, when this part of the world was open grassland grazed by mammoths, woolly rhinos, and bison. What survived into our century is remarkable. Yuka preserved skin, hair, muscle, soft tissue, and cranial material. Patches of reddish fur still cling to the hide. The lower legs are so intact that, with the skin peeled back, the muscle underneath looks almost fresh. Here's the distinction worth holding on to, because it shapes everything in this story. A fossil bone is not really bone anymore. Over tens of thousands of years, minerals replace the original tissue, turning it into stone. A frozen carcass works differently. The cold simply stops decay. It does not replace anything, it pauses it. So when you study Yuka, you are not studying a stone copy of a mammoth. You are studying actual mammoth, biology and all, held at the moment of death. That includes the brain. Yuka's skull preserved a mammoth brain with its folds visible, and even traces of blood vessels still readable on the surface. In modern terms, this is the difference between having a photograph of an engine and having the engine itself, still bolted together. But permafrost is a fragile kind of vault. It is a natural freezer, and a natural freezer fails when the power goes out. Thaw conditions in Siberia are unstable, and a carcass exposed by melting ground will rot within seasons if nobody intervenes. After Yuka was recovered, the carcass was moved into dedicated ice storage to keep it frozen because the alternative was watching tens of thousands of years of preservation disappear in a single warm summer. Now, one correction about Yuka itself. For years, the specimen was described as female, often nicknamed she in press coverage. Recent genetic work overturned that. The molecular evidence identifies Yuka as male. It is a small detail, but it shows how even a body this complete can mislead us until we read it at the chemical level. And reading Yuka at the chemical level is where the modern breakthrough arrives. You've probably heard of ancient DNA. DNA is the inherited code, the blueprint, the instruction set an animal is born with. Ancient RNA is a different thing entirely. RNA is the working copy. It is the molecule that carries instructions from the genes to the cell's machinery. And crucially, it reflects which genes were switched on at a given moment. DNA tells you what an animal could do. RNA tells you what it was actually doing. In 2025, a research team led by Amelia Marmol Sanchez of the University of Copenhagen, working with paleogeneticist Love Delen of Stockholm University, did something the textbook said was impossible. They extracted and sequenced RNA from Yuka's front leg muscle. This is the oldest RNA ever recovered by a wide margin. RNA was thought to be far too fragile to last more than hours or days after death. Yuka's RNA lasted nearly 40,000 years. What did it show? The recovered molecules coded for proteins involved in muscle contraction and metabolic regulation under stress. Out of more than 20,000 protein-coding genes in the mammothid genome, only a fraction were active in that tissue, and the pattern is exactly what you would expect from muscle. The team described it as measuring the last minutes or hours of the animal's life. There are hints of stress signals and muscle metabolism near death, the cellular equivalent of catching an animal mid-effort. Yuka's hide also carries trauma marks. Some researchers interpret them as damage consistent with a cave lion attack, but the cause of death stays unresolved. We have a body, we have wounds, and we do not have a verdict. Honest research leaves it there. Now we need to clear up a legend because Yuka is constantly confused with another mammoth. You may have heard the story of mammoth liquid blood dripping from a frozen carcass. That did happen, but not with Yuka. It happened with a different specimen, a mammoth nicknamed Buttercup, found on the Malolyakovsky Island site. The liquid blood story got attached to mammoth cloning headlines and then drifted, in popular retellings, onto Yuka, because Yuka was the famous one. Cloning labs did receive mammoth samples for study, but they were sample recipients, not resurrection factories. Nothing about Yuka brings a mammoth back to life. And every time a researcher cuts a sample from a specimen like this, the specimen gets smaller. Destructive sampling is permanent. The vault that keeps Yuka frozen is not hiding a conspiracy. It is cryogenic protection for a body that would otherwise decay. So Yuka sits in cold storage, a rare case where Ice Age tissue still carries biological messages, which raises a question we will keep meeting. If a frozen mammoth can be protected this carefully, what happens when the specimen is not a mammoth at all, but a set of broken bones that might rewrite the entire story of how humans reached the Americas? Number 2. The Cerutti Mastodon specimens. In 1992, a freeway expansion project in San Diego, California, cut into the ground and exposed a deposit of old bones. Paleontologist Richard Cerutti, working ahead of the construction crews, recognized what was emerging. These were mastodon remains, limb bones, tusk fragments, molars, and, scattered among them, large stone cobbles that seemed out of place. For decades, the site sat quietly in the collections of the San Diego Natural History Museum. Then, in 2017, a team published a date that detonated across the field of American prehistory. The Cerutti Mastodon site, they said, was about 130,700 years old, with an uncertainty of plus or minus 9,400 years. To understand why that number is explosive, you need the baseline. The broadly accepted evidence for humans in the Americas clusters around 15,000 to 20,000 years ago. Some sites push a little earlier and remain debated. The Cerutti claim does not push the timeline a little earlier. It pushes it back by more than 100,000 years. If correct, it would mean some kind of tool-using hominin was in California before our own species had even spread across Eurasia. The dating method at the core of the claim is uranium series dating, applied to the bone itself. Uranium absorbed into bone decays into thorium at a known rate, and the ratio acts as a clock. That clock is what produced the 130,000-year figure. The behavioral argument rests on the bones. Some mastodon limb bones show spiral fractures, the kind of break that happens when bone is broken while still fresh, still containing its organic content, rather than cracked dry and brittle long after death. The team also reported percussion damage and impact notches, marks of something heavy striking the bone. They proposed a tool system. Certain large stones served as hammerstones, others as anvils, and the mastodon limbs were broken on them, possibly to extract marrow. They described some of the stones as hydraulically anomalous, meaning the cobbles were too large to have been carried into that fine, low-energy sediment by water alone. Something else put them there. Now the weaknesses stated plainly. No human bones were found at the site. No classic chipped stone tools, no recognizable spear points or scrapers were found either. The argument is built on association. Bones, stones, and damage occurring together. Association is not the same as direct human remains. This is where the field splits. Critics argue the breakage is natural. Heavy construction equipment passed over the deposit. Sediment shifts, the weight of overlying earth, and ordinary geological processes can fracture bone and concentrate stones. They argue the team misread taphonomy, the science of what happens to remains after death, and saw butchery where there was only geology. Proponents answer that carbonate crusts formed across some of the fracture surfaces, and crusts take time to grow. If the breaks were ancient enough to be coated in mineral crust, they argue, a bulldozer in 1992 did not cause them. They also point to experimental work where researchers deliberately broke elephant bone with stone tools and produced fracture patterns and notches comparable to what Cerutti shows. The Mastodon material has been part of the San Diego Natural History Museum's public exhibit history, and the current restriction on access is best understood as research access control, rather than total invisibility. Museums hold contested specimens carefully precisely because the dispute is unresolved. Every researcher who handles them could damage the evidence the next team needs. So the Cerutti question stays open. Was this a genuine human site, the oldest in the Americas by a staggering margin? Or was it a geological trap, a coincidence of bones and rocks that invited a century-shifting interpretation it could not support? The honest answer today is that we do not know. And if a contested site can hang in limbo like this, consider what happens when the specimen is not ambiguous bones, but a child deliberately buried who walked straight into a war over whether two human species ever became one. Number 3. The Lapedo Child. In the Lapedo Valley of central Portugal, there is a rock shelter called the Abrigo do Lagar Velho. In 1998, excavators there uncovered a small human skeleton, and it was given the formal name Lagar Velho 1. It is better known as the Lapedo Child. The skeleton belonged to a child of about 4 or 5 years old. The body had been placed in a deliberate burial stained with red ochre in a context belonging to the Upper Paleolithic. This was not a casual abandonment. It was a grave, prepared with care, in a period when our species was already spread across Europe. The age of the burial was first reported as around 24,500 years. More recent work revised that to a range of roughly 27,780 to 28,550 calibrated years before present. The revision came from a refined radiocarbon technique called compound-specific dating, which isolates hydroxyproline, an amino acid specific to collagen, the protein in bone. Why go to that trouble? Because collagen in old bone is easily contaminated by younger carbon, and an early complication at this very site was root penetration, plant roots growing through the burial and introducing material that throws off the clock. By dating only the hydroxyproline, researchers strip away much of the contamination and read a cleaner signal. The reason the Lapedo Child became a scientific flashpoint is its anatomy. The original interpretation, led by Eric Trincaus and the Portuguese archaeologist João Zilhão, described the skeleton as a mosaic. The child had a chin that looked essentially modern, a feature associated with Homo sapiens, but it also had body proportions, particularly in the limbs, that researchers described as more archaic, closer to what is seen in Neanderthals, who were adapted to cold climates with shorter, stockier builds. A mosaic of modern and Neanderthal-like traits in a single body, in Portugal, several thousand years after Neanderthals were thought to have vanished. The interpretation was that this child descended from a population that carried both ancestries. It is important to be precise here. The claim was population admixture, the mixing of groups over generations, not necessarily a first-generation hybrid with one Neanderthal parent and one modern human parent. At the time, this landed in the middle of a fierce debate. One camp held a strict replacement model, modern humans spread out of Africa and replaced Neanderthals with little or no interbreeding. The other camp argued for genuine mixing. The Lapedo Child was offered as fossil evidence for mixing, and it was contested hard, because in the late 1990s, the genetic tools to settle the question did not yet exist. Then they did. Ancient DNA, recovered first from Neanderthal fossils and later compared against living people, confirmed that non-Africans today carry a measurable percentage of Neanderthal ancestry, generally a low single-digit fraction. Interbreeding happened. It is no longer a question. That genetic confirmation changed the emotional temperature of the debate. The shock of the Lapedo Child faded because a mosaic individual is no longer extraordinary once you know the two populations mixed. But the genetics did not erase the fossil's value. The bones remain a rare, well-documented Upper Paleolithic child burial, and the anatomical debate over how to read those specific proportions continues among specialists. Juvenile remains are especially fragile. A child's bones are thinner, less mineralized, and easier to damage than an adult's. The Lapedo Child holds national treasure status in Portugal and is curated in Lisbon, and for public interpretation, replicas are generally used because a cast can be handled and displayed without risking the original. There is also a quieter ethical dimension. This is the body of a small child, buried by people who grieved. Displaying such remains is handled with sensitivity. The Lapedo Child stands as evidence that human categories were never as cleanly separated as the old textbooks drew them. And if the boundary between human groups was that blurry, then the next specimen pushes the problem further back into the Middle Pleistocene, where a single skull has spent more than 60 years resisting every attempt to say exactly what it is and exactly how old it is. Number 4. The Petralona Skull. In 1960, in the Petralona cave near Thessaloniki in northern Greece, a skull was found. It was a cranium, a brain case with the face attached but missing its lower jaw. From the start, it was clearly something archaic, a Middle Pleistocene human, but not a member of our own species. The skull is robust in a way modern skulls are not. It has a heavily built cranial vault and a powerful face. The cranial walls are thick, the brow ridges, the bony ridges above the eye sockets, are pronounced. It has a broad forehead, large zygomatic arches, the cheekbone structures, and a strong maxilla, the upper jaw. In modern terms, this is a heavy-duty build. A skull engineered for stresses our own thin-walled crania are not, and from the moment it was found, it was a problem. The trouble began with the find spot itself. The records of exactly where in the cave the skull lay and in what sediment were poor. Without a secure find spot, you cannot date the layers around it, and the dating debate spiraled. Over the decades, proposed ages for the Petralona skull range from about 170,000 years all the way to 700,000 years. Associated cave fauna suggested broadly a Middle Pleistocene age, but that is a wide window. In 2025, a team led by Christophe Falguer of the Institut de Paléontologie Humaine in Paris, working with the Natural History Museum's Chris Stringer, among others, attacked the problem from a new angle. Instead of dating the uncertain sediments, they dated the calcite. Calcite is the mineral that forms when mineral-rich water seeps through a cave and dries, leaving crusts on whatever surface it touches. A layer of calcite had grown directly on the skull itself. Using uranium series dating on that crust, they produced a result: a minimum age of 286,000 years, plus or minus 9,000. Here is the key logic. The calcite grew on the skull after the skull was already in the cave, so the calcite date tells you the skull is at least that old. It is a minimum age, not a death date. The team estimated the skull itself is probably around 300,000 years old, possibly older. The researchers did one more careful thing. They compared the calcite on the cranium to calcite from the cave wall in the chamber called the mausoleum, where the skull was reportedly found. The two did not match. They formed during different phases of the cave's history. That mismatch matters because it means the simple story of where the skull came from is still not settled, even now. What was this individual? The Petralona skull is generally classified as Homo heidelbergensis, or a closely related archaic human of the Middle Pleistocene. It is not modern Homo sapiens. It is not a classic Neanderthal either, though it shows some features that point toward the lineage leading to Neanderthals. Researchers comparing it to the Kabwe skull from Africa have noted similarities, hinting that Middle Pleistocene populations in Europe and Africa may have been more connected than once assumed. The honest position, stated by the researchers themselves, is that the skull cannot be firmly assigned to a single named ancestor. There is also a custody dimension. The skull is held in Greece, and Greek institutional control over it has, at times, intensified the controversy, with disputes over study access. Casts are used for controlled presentation, and the original is tightly managed. The vault around the Petralona skull is partly scientific protection and partly a legal and cultural question of who controls a national fossil. So the skull asks two questions at once: how old is it? And who owns ancestry? Hold that second question because it is about to take over completely. But first, a different kind of dating story, one where the new number did not make a specimen more dramatic, it made it less. Number 5. The Liujiang Skeleton. In 1958, in the Guangxi region of southern China, inside a cave called Tongtianyan, workers found human bones. What emerged was one of the most complete early Homo sapiens skeletons ever found in East Asia. The Liujiang skeleton includes cranial material, teeth, and postcranial remains, meaning bones from below the skull, the limbs, and the rest of the body. That completeness is rare and valuable. Across human evolution research, most fossils are fragments: an isolated skull, a single jaw, a scatter of teeth. A near-complete skeleton lets researchers study an individual as a whole organism, comparing skull shape, dental traits, and limb proportions in one person. For decades, Liujiang carried a dramatic claim. Some studies proposed very ancient ages for the skeleton, in some estimates up to 227,000 years. If that were true, it would mean modern humans were in southern China far earlier than the standard out-of-Africa timeline allows, which places the major dispersal of our species out of Africa well after that. A 200,000-year-old modern human in China would be a serious challenge to the accepted model. But there was a problem, and it is the same problem we saw at Petralona. The skeleton came out of a cave in 1958, and the provenance, the precise record of which layer it came from, was weak. Unclear stratigraphy can mislead a field for decades because you end up dating sediments that may have nothing to do with the fossil. In 2024, an international team including Junyi Ge, Song Xing, and Qingfeng Xiao, with contributions from Griffith University's Michael Petraglia, redated the skeleton properly. They used 3 independent methods. Uranium series dating was applied directly to the human fossils. Radiocarbon dating was applied as a second method. And optically stimulated luminescence, a technique that measures how long sediment grains have been buried away from light, was applied to the fossil-bearing sediments. The reason to use 3 methods is straightforward. When independent clocks agree, the uncertainty drops sharply. They agreed. The new age for the Liujiang skeleton is approximately 33,000 to 23,000 years ago, not 227,000. Around 30,000. That revision quietly dismantled the dramatic storyline. Liujiang no longer supports a modern human presence in China beyond 130,000 years ago. The co-author, Qingfeng Xiao, put the principle bluntly. A human fossil without a reliable age is essentially useless, and the younger date finally places this individual in the correct position on the human family tree. And that position is still important. The new date aligns Liujiang with other late Pleistocene Homo sapiens fossils in China, such as those from Tianyuan Cave and Zhoukudian Upper Cave, showing our species was widespread across Eastern Asia by this period. Liujiang remains a rare, near-complete East Asian early modern human skeleton. Its cranial shape, its teeth, and its limb proportions are valuable for understanding population variation among early modern humans, not just for migration theory. The skeleton is held under Chinese national heritage custody, with access managed for specialist review. Liujiang shows something important about this whole field. A better date can erase a dramatic claim without erasing a specimen's worth. The fossil did not change. Our confidence about time did. Now, we move to a case where dating did the opposite, where it narrowed in on the final chapter of an entire human species. Number 6. The Ngandong, or Solo, skulls. Along the Solo River on the Indonesian island of Java, the land is built up in terraces, old riverbank levels left behind as the river carved downward over time. In one of those terraces, at a site called Ngandong, excavations between 1931 and 1933 uncovered an extraordinary group of human fossils. The haul was 12 calvaria and 2 tibiae. A tibia is a shin bone. A calvaria is a skull cap, the upper brain case, without the full facial skeleton. So this was a concentration of partial skulls and a couple of leg bones, and they belonged to Homo erectus, the long-lived human species that preceded our own across much of the Old World. The bones were found in a bone bed roughly 20 meters above the present level of the Solo River, sitting in those terrace deposits. River terrace deposits are a double-edged thing for science. They are evidence because they trapped and preserved the bones. But they are also a dating challenge, because river systems move material around, mixing older and younger sediment. What made Ngandong famous is its place in time. These were presented as among the youngest known Homo erectus anywhere. To appreciate that, consider the span involved. Homo erectus reached Java more than 1.5 million years ago. The species had an extraordinarily long run, far longer than our own species has existed so far. Ngandong appeared to capture its final phase, but the precise age was uncertain for a long time, with estimates scattered widely. Then, in 2019, a study published in the journal Nature applied a rigorous solution. The team gathered 52 radiometric age estimates from the site and surrounding deposits and combined them using Bayesian modeling, a statistical method that integrates many separate dates with all their individual uncertainties into one coherent age range. The dates came from multiple techniques: uranium series dating, luminescence dating, argon-argon dating, which is used on volcanic material, and combined uranium series and electron spin resonance approaches. The refined result placed the Ngandong Homo erectus fossils at roughly 117,000 to 108,000 years ago. That is a striking number. It means Homo erectus was still alive on Java when modern humans were already established elsewhere in the world. But late survival is not the same as direct contact. The Ngandong dates do not prove that these Homo erectus populations ever met Homo sapiens face to face. What the dating supports is the idea of island Southeast Asia as a possible refugium, a pocket of the world where an older human species held on after it had disappeared elsewhere. As for how 12 skull caps ended up concentrated in one bone bed, the leading explanation is flood processes. Periodic flooding of the Solo River likely gathered the remains and deposited them together, which also explains why the sample is skull caps and leg bones rather than complete skeletons. The Ngandong material is curated in Bandung, Indonesia, under controlled conditions, which is best read as careful curation of fragile, scientifically critical fossils. Ngandong is the last recorded chapter of a species that lasted well over 1 million years, and from the end of an ancient lineage, we turn to something that complicates a different assumption entirely, the assumption that Ice Age people lived simple lives. Because the next site contains 2 children buried with a level of wealth that is genuinely hard to comprehend. Number 7. The Sungir Children. Near the city of Vladimir in Russia, about 200 kilometers east of Moscow, lies the Upper Paleolithic site of Sungir. It is one of the richest Ice Age burial sites ever excavated, and its most famous feature is a double burial of 2 children. The 2 children were placed head-to-head in a single grave. Age estimates in various reports put them at roughly 10 and 12 years old. Their sex has been assessed differently across different studies and remains a point of discussion. What is not in dispute is the staggering quantity of material buried with them. The grave contained more than 10,000 beads carved from mammoth ivory. Some broader descriptions of the Sungir burials put the total bead count across the site in the region of 13,000. Now think about what a single bead represents. Each one had to be cut from ivory, drilled, shaped, and polished. Researchers estimate the labor in the thousands of working hours. These beads were sewn onto clothing, so the children were dressed head to foot in garments covered in handmade ivory. The beads are only the beginning. The burial included more than 20 armbands. It included around 300 pierced fox teeth, each tooth drilled so it could be attached, likely to clothing or headgear. There were ivory spears, carved artifacts, deer antlers, and other grave elements. Among the most remarkable objects were long lances made from mammoth ivory, straightened from naturally curved tusks, a genuine feat of working organic material. And laid across the children's chests were 2 human fibulas, leg bones from other individuals placed there deliberately. In modern terms, this is not a simple grave. It is closer to a state funeral. The labor invested is enormous, and labor on that scale is evidence of social complexity. It tells us the Sungir community could organize, sustain, and direct large amounts of skilled work toward a ritual purpose. That raises hard questions about these 2 children specifically. Why them? Children generally have not had time to earn status through their own achievements, so a burial this rich points toward possibilities like inherited status, where rank passed down through families, or a special ritual role assigned to the children, or perhaps care connected to disability, or symbolic kinship of some kind. Sungir does not let us choose between these with certainty. It only tells us that whatever the reason, it mattered enormously to the people who dug the grave. Ancient genome studies of Upper Paleolithic populations, including individuals from Sungir, have added another layer. The genetic evidence points to wider mating networks and relatively low levels of inbreeding, suggesting these groups were not tiny isolated bands but were connected across considerable distances, deliberately maintaining links with other communities. The Sungir remains are curated primarily in the Vladimir Suzdal Museum Reserve. For public display, models and replicas are generally used, both to protect the originals and because juvenile skeletons are especially vulnerable. Sungir stands as direct evidence that Ice Age societies had deep ritual structure, social hierarchy, and symbolic life. The old picture of Paleolithic people as purely survival-driven does not survive contact with this grave. So far, every vault we have discussed has been about preservation, dating, or contested science. The final specimen changes the terms completely. It is locked away not because of fragility or doubt, but because the question of who has the right to it was finally answered, and the answer was not the museum. Number 8. Mungo Man, also known as Lake Mungo 3 or LM3. In the Willandra Lakes region of New South Wales, Australia, there is a dry lake bed called Lake Mungo. In February of 1974, the geologist Jim Bowler, examining the eroding landscape, found human remains there. The remains received the designation Lake Mungo 3. The individual became known as Mungo Man. Mungo Man is approximately 42,000 years old, and the burial was not simple. The body had been laid out and coated with red ochre. Ochre is not ordinary sediment. It is a pigment, gathered and prepared, used across human cultures as a ritual material. The presence of ochre transforms this from a death into a ceremony. Mungo Man represents one of the oldest known ritual burials anywhere outside Africa. When their remains were found, they were removed and taken to the Australian National University in Canberra for study. By the standards of 1974 archaeology, that was routine. By later standards, that removal became deeply contested because Mungo Man is not only a scientific specimen. To the Aboriginal communities of the region, he is an ancestor, and he had been taken from his country without their consent. The traditional owner groups connected to the Willandra Lakes region are the Muti, the Pakanti, also written Pakanchi or Barkinji, and the Ngyampa peoples. For decades, these communities campaigned for the return of Mungo Man and the other ancestral remains held in institutions. That campaign succeeded in stages. In 2015, the Australian National University formally repatriated the remains. Mungo Man was then housed for a period at the National Museum of Australia. And in 2017, Mungo Man was returned to country, brought back to the Willandra Lakes landscape together with 104 other sets of ancestral remains. The concept that governs his care now is a keeping place, a site where ancestral remains are held under the cultural authority of the traditional owners themselves. Scientific research can still happen, but only through the joint permission of the descendant communities. This is a very different model from a museum vault. In a museum, the institution decides. Here, the descendants decide. It is worth being precise about what the restriction means. Mungo Man is not hidden because of secrecy. The restriction reflects cultural authority. The communities whose ancestor he is now hold the decision over who may study him and how. The earlier removal happened in an era when community consent was simply not sought, and the repatriation is best understood as a correction of that. Mungo Man is the one specimen on this list that has moved beyond replica logic, beyond the museum framework entirely. The public lost a certain kind of access to him, but not because something was being concealed. It was because the question of ownership was reconsidered and answered differently. That brings us to a larger question that runs underneath all 8 cases. What does a vault actually mean in paleoanthropology? The word vault is doing a lot of work, and it is worth defining broadly. A vault might be a freezer holding a frozen carcass. It might be a climate-controlled cabinet in a laboratory. It might be a museum's reserve collection off the public floor. It might be a cultural keeping place. These are very different things, and lumping them together as locked away hides what is really going on. Start with fragility. Many original specimens are simply too delicate for constant display. Light, vibration, and handling all cause cumulative damage. So museums make casts, replicas, and increasingly three-dimensional prints. A good cast reproduces the external shape of a bone with high accuracy, and visitors look at it without ever knowing. Then there is environmental control. Bone contains collagen, and collagen degrades faster in fluctuating humidity, so collections control humidity tightly. Frozen tissue like Yuka's needs stable sub-freezing temperatures, which is why such a specimen cannot sit in an ordinary glass case. Ultraviolet light breaks down organic material, so it is filtered or excluded. Handling introduces skin oils, microbes, and abrasion, all of which contaminate and erode. For contested specimens, there is also chain of custody, the documented record of who has handled a specimen and when. This matters when a fossil's authenticity or context is disputed, and there is the question of destructive sampling. Some analyses, including genetic and molecular work, consume part of the specimen. With rare material, a few samples can exhaust what exists. So institutions form committees to weigh sampling requests. This is why public display and research access are different things, governed by different rules. and it is why a cast can sometimes be better for public teaching than a damaged original. The cast shows the complete clean shape. Non-destructive technologies help close the gap. CT scanning images internal structure without cutting. Micro-CT resolves fine internal bone detail, and surface scanning generates the digital models used for replicas and shared with researchers worldwide. Specimens are also loaned between institutions, and genetic testing or export often requires legal permits, while ancestral remains carry cultural restrictions on top of all of this. The point is simple. Vaults preserve evidence for a future technology. The Yuka RNA result happened because the specimen was protected long enough for the method to be invented. Secrecy is usually far less important than survival, which leads directly to the issue that has surfaced in nearly every section: dating and why one number can overturn a story. Dating is the skeleton of the entire narrative. Get the date wrong, and every interpretation built on it collapses. There are two broad approaches. Direct dating measures the specimen itself. Associated context dating measures something near the specimen, like the surrounding sediment. In the danger, there is obvious. You might be dating material that has nothing to do with your fossil. Each method has a domain. Radiocarbon dating works on organic material, but it has a practical ceiling, generally around 50,000 years, beyond which too little of the radioactive carbon remains to measure. Uranium series dating works well on cave deposits and on fossils, using the decay of uranium into thorium. When applied to a calcite crust, it gives a minimum age, because the crust forms after the specimen is already in place. Optically stimulated luminescence dates the last time sediment grains were exposed to sunlight, which times their burial. Argon-argon dating works on volcanic material. Electron spin resonance, often combined with uranium series, is applied to tooth enamel and similar materials. And Bayesian modeling is the statistical tool that combines many separate dates into one range. You have seen each of these do real work. Cerutti's 130,700 year date, with its 9,400 year uncertainty, is the high-stakes example. A single number that, if confirmed, rewrites a continent's prehistory. Petralona's 286,000 year minimum age shows how a date can be firm and still incomplete. Liujiang's redating shows correction in action. Ngandong's 52-date Bayesian model shows synthesis, many uncertain numbers combined into a confident range. Contamination is the constant enemy. Roots, minerals, and handling all introduce foreign material. This is why provenance, the secure record of where a specimen came from, can matter more than how beautifully it is preserved. A flawless skull with an unknown find spot can be nearly useless for dating. And this is why responsible researchers report confidence ranges plus or minus a figure rather than a single magic number. New methods keep rewriting old narratives. In this field, time itself is often the most contested artifact. It is worth stepping back to see why these 8 specimens are valuable in genuinely different ways, because they represent 4 different kinds of evidence: bones, soft tissue, molecules, and stone. Frozen tissue is the rarest preservation category, and Yuka is the example. From such tissue, you can pull DNA, the inherited sequence, RNA, the record of active gene expression, and proteins, which are more durable molecular traces than either. You can also study histology, the structure of the tissue itself under magnification. Mammoth muscle and mineralized fossil bone are simply not the same kind of evidence. Then there is bone as a record of mechanics. The Cerutti debate turns on fracture mechanics. A fresh bone, still organic, breaks in a spiral. A dry old bone cracks straight and brittle. Percussion marks and impact scars are read the same forensic way. Cranial morphology, the shape of the skull, is the evidence in Petralona. Dental traits carry their own signal, as in Liujiang. Postcranial anatomy gives body proportions, which is exactly what made the Lapedo Child controversial, and calvaria, partial skulls, are the evidence at Ngandong. The third category is social and symbolic evidence. Grave goods speak to social structure. Ochre speaks to ritual. The Sungir beads speak to labor, thousands of hours of it. Isotopes preserved in bone and teeth can, in the right circumstances, speak to diet and movement, though not every specimen yields such data. And genome studies can reconstruct ancient social networks, as they did at Sungir. One specimen can therefore support multiple disciplines at once. Yuka is biology and chemistry, Cerutti is geology and archaeology, Sungir is anthropology and genetics. Biology, geology, technology, and ritual all converge in the same drawers. This also explains, more concretely, why the public usually sees replicas instead of originals. Most likely, the next time you look at a famous fossil in a museum, you are looking at a cast and do not know it. The reasons stack up. Replicas protect fragile originals from light exposure. They reduce the risk of vibration and transport damage. There are insurance and security concerns because famous fossils carry real theft risk and real monetary value. There is damage risk from crowds and from the climate swings a busy gallery experiences. Juvenile skeletons like the Lapedo Child and the Sungir Children are especially vulnerable. Frozen carcasses cannot survive in ordinary display cases at all. Sacred or ancestral remains may not be shown, out of cultural respect, and contested specimens may be held primarily for research while their disputes are unresolved. Educational value and research value are different things, and institutions balance them. Three-dimensional scanning offers a compromise, feeding both digital archives that researchers worldwide can access and the physical replicas that fill display cases. Sampling committees ration access to the original material. Interestingly, media access is often stricter than academic access, because a researcher with a documented project and institutional backing is a known quantity. You can see the pattern across our cases. Petralona is shown as a cast while the original is tightly held. The Lapedo Child is interpreted for the public through replicas. Sungir is presented through models. And Mungo Man sits beyond replica logic entirely because his case is not about fragility, but about sovereignty. That word, sovereignty, points to the last theme, the one that turns these specimens from objects into something more complicated: ownership, nationalism, and indigenous authority. Begin with the genuinely difficult question: who owns a human ancestor? It is worth separating categories here. A fossil of an extinct species, an artifact made by human hands, and the ancestral remains of a known descendant community are not the same kind of thing, ethically or legally, even if all three end up in a museum. Several systems of control overlap. National heritage laws can declare a specimen the property of a state. Museum accession and custody govern how an institution holds material. Excavation permits and the rules of legal recovery determine who could dig in the first place. And there can be tension between a university that did the original research and the state that claims ultimate ownership. Our cases map this directly. Petralona is the ownership conflict example, a fossil entangled in national and institutional control. Mungo Man is the repatriation example. Liujiang sits under national research custody in China. Sungir sits in a Russian state museum reserve. Source countries restrict export to keep heritage within their borders. Destructive testing requires formal permission. And for ancestral remains, cultural consent is its own requirement, separate from any law, because descendant communities may simply decline scientific access, as is their right. There is real scientific frustration in this. Researchers want access. Restrictions slow discovery. That frustration is understandable, and it does not have to be dismissed in order to also recognize that many older excavations proceeded with no community consent at all in eras when consent was not considered. Repatriation is a modern correction to that history. A specimen can shift over time from being treated as an object to being recognized as an ancestor. The access rules around these specimens are not arbitrary. They reveal who holds power over the past. A vault, in that sense, is not only a physical space, it is also a political and legal boundary. Given all of that, it is worth laying out, fairly, the competing ways to interpret why these specimens are locked away. There are at least three. The mainstream interpretation is the simplest. Restricted access is mainly about preservation and ethics. Vaults are conservation infrastructure, nothing more sinister. On this view, the skepticism around Cerutti is appropriate because an extraordinary claim. Humans, in America, 130,000 years ago, genuinely does require extraordinary proof. The Liujiang redating is a correction, not a suppression. Yuka is a biomolecular archive valuable for its RNA, not a shortcut to cloning. And Mungo Man is an ancestor under the rightful authority of his community. This interpretation treats the whole pattern as ordinary, careful science and ethics. The archaeological and historical interpretation is more textured. It says the vaults reflect the messy history of how these specimens were discovered. Early excavations kept poor records. The colonial era extracted fossils and remains from around the world with little regard for local people. Institutions competed, and rivalries shaped which specimens were studied and how. On this view, Petralona is a fossil shaped by national and academic conflict as much as by science. The Lapedo Child is a fossil caught in a pre-genomic identity debate, argued fiercely before the genetic tools existed to settle it. Ngandong is a colonial-era discovery later reinterpreted by better dating. And Sungir is read as evidence that Ice Age societies had genuine social hierarchy. This interpretation does not allege conspiracy. It says the vaults carry the fingerprints of history. The fringe, or speculative, interpretation makes a stronger claim that locked specimens hide paradigm-changing truths. It points to Cerutti as evidence of suppressed early Americans, a discovery the establishment will not accept. It points to mammoth tissue as proof that cloning is closer than scientists admit. It points to Liujiang's old large dates as evidence of an ignored Asian origin for modern humans. The intellectually honest way to treat this view is with curiosity rather than contempt, but also with a clear standard. It requires testable evidence, and so far it does not provide it. There is a real and interesting observation buried inside it, though. Restriction genuinely can create suspicion, even when plain conservation is the actual reason. When originals are inaccessible, secrecy narratives grow to fill the gap. That is a fact about human psychology, and it is worth understanding rather than mocking. Weigh the three together, and the conclusion is not dramatic. The strongest mystery here is not conspiracy. It is uncertainty under protection. These specimens are genuinely unresolved, and they are genuinely protected, and those two facts together generate most of the intrigue. So pull the 8 cases into one frame. Why can't the public see them? Yuka belongs with biomolecular preservation, a body kept frozen so its molecules survive. Cerutti belongs with contested behavioral evidence, bones and stones that may or may not record human action. The Lapedo Child belongs with hybridization and burial ethics. Petralona belongs with the tangle of age, taxonomy, and ownership. Liujiang belongs with redating and scientific self-correction. Ngandong belongs with extinction and the late survival of an older species. Sungir belongs with ritual wealth and the burial of children. Mungo Man belongs with repatriation and ancestral sovereignty. One thread runs through every one of them. Each specimen changed because a new method arrived. Yuka changed when RNA sequencing reached back 40,000 years. Petralona changed with calcite uranium series dating. Liujiang changed with 3 combined dating techniques. Ngandong changed with a 52-date Bayesian model. The vault, in each case, bought time. It kept the specimen intact long enough for the technology to catch up. That is why the surface appearances are so misleading. Preservation can look like secrecy. Scientific caution can look like suppression. Cultural authority can look like a denial of access. None of those appearances is the reality. And critically, not all 8 specimens are locked away for the same reason. Yuka is about temperature. Cerutti is about unresolved debate. Mungo Man is about sovereignty. Treating them as one phenomenon is the mistake. What the evidence rewards is precision over myth and a tolerance for uncertainty as a normal part of honest research. Some specimens really are too fragile to sit behind glass. Some are too sacred, and some are too dangerous to careful interpretation, too easily misread to be handed to the public before the science is settled. The unseen originals are still shaping human history. They are still being dated, sampled, scanned, and argued over, out of public view. The vault is not the end of the story. It is the place where the next version of the story is being written, and where it waits for the method that has not been invented yet.